OsteoSignal Episode 11. This is OsteoSignal with Neal D. O'Neill, a podcast exploring how your body's natural design connects with osteopathic science, translating complex signals into practical insights so you can truly thrive.
Welcome to OsteoSignal with Neal O'Neill, and with me today is...
Hey there, I'm Brent. I'm here representing the average Joe, the layperson here. Just asking Neal questions about what he's going to present to us and how we can make the most sense and use out of it.
Yeah, perfect. So today I wanted to talk about, there's all sorts of research coming out and a lot of information about peptides and brain health and things like that. So what we want to do is kind of delve into that. And I'm an osteopathic manual therapist. I work on people to make them work better. And so then understanding and some of this research that's coming out and how it can be the most useful for patients, I like to give a lot of background information, a lot of physical health parts, like what do I do that helps or hinders or alters the whole function of some of these things. Because as we all know, not everyone has the same response to medical intervention. And in a lot of ways, what used to be the area of nutraceuticals is now becoming more and more the purview of the pharmaceuticals. So
could you highlight the distinction real quick between nutraceuticals? Is that just things you find in nature that aren't, processed by humans? And what are pharmaceuticals?
So pharmaceuticals are definitely highly chemically synthesized compounds. Now they may be found in nature, but they are definitely synthesized in a lab, and they're made, right? There's a chemical process that is gone through to make these very pure or highly pure chemicals and for their medicinal use. Now, there's a lot of things that, so of course, like painkillers or antibiotics or things like that are something that we all know, but there are a lot of other substances out there. So for example, like say we're going to take a statin, we might get Lipitor, and that would be a pharmaceutical because it is a derivative of red yeast rice. And they've taken the compounds in red yeast rice and they've synthesized them, or at least taken them and made a very pure compound out of that so that it has just the effect thereafter. They don't have anything else in there. Whereas a nutraceutical might be that an individual goes to the health food store and picks up red yeast rice. That's not nearly as powerful as a statin, but has the same effect or has a similar effect with dosage and stuff. And so since that falls below the grade of what the medical community and the pharmaceutical community is really interested in, the government regulation differs. On pharmaceuticals, there's high government regulation, or at least there's supposed to be. There's supposed to be regulation as to how it's made, how pure it is. And of course, that does also speak to like if something goes wrong, who can be sued, how much can they be sued for? So a pharmaceutical has a much higher level of responsibility in that way. Whereas a nutraceutical is like, hey, look, this is just a food supplement or a supplement. It's a food product. So the FDA does not, the Food and Drug Administration does not oversee, the quality of it or the use of it or where it's sold or who sells it.
Or claims of its effectiveness.
Right? And
I see
in
those
fact, you
labels all
can't
the
claim.
time.
You can't claim that it does anything. You could say, well, you can say, well, people have said that they've had this, but you can't say we've found in clinical studies because again, that level of medical or clinical study has not been performed on these nutraceuticals, or maybe it has, but it hasn't been accepted by the FDA because it falls below their level of what they want to, they want to oversee, right? I mean, of course they can't oversee everything, and there has to be room for people to live their lives. So for example, whole milk or milk that has not been homogenized or cooked, right?
Pasteurized.
So it hasn't been pasteurized. That sort of thing, like the FDA is like, we don't really, in the past, they've said no way, only in very specific circumstances. And of course, things are changing. And so that's kind of changing. So even just raw milk is a nutraceutical. But it's on that edge of what do we accept and what do we not accept. So there you have it. So there's some gray area there as well.
All right. Thanks for taking that detour.
Yeah, sorry.
Appreciate it. All
So
right, it's all
so
good.
it kind of tells us nutraceuticals, pharmaceuticals. So and all the peptides that we're seeing out there, there's a lot of use of peptides, and in the past, those have all been nutraceutical. The issue, though, is that things like human growth hormone or insulin or GLP-1s, the Ozempics, those things had been in the realm of nutraceutical, but I think we can all agree that insulin maybe needed to be much more pure, much more regulated, much more overseen, and so that has fallen under the FDA's purview. So there's a peptide that has been under the purview of the FDA. However, there's a lot of other peptides that were not under the FDA's purview. However, and I think it actually has something to do with the use of AI. In the past, pharmaceuticals used MDs, medical doctors, and then later PhDs, and say, for example, in biochemistry, to find chemicals that they thought would have a therapeutic effect on, for certain medical conditions like blood pressure, like weight, type 2 diabetes, et cetera, et cetera. And in the past, it was, a lot more research and more human capital doing that. But with and this is my opinion, I think with the kind of the onset of AI, those people have started using AI, and then a lot more of the information that's out in the world, for example, back to these nutraceuticals that haven't been overseen as much as pharmaceuticals have, but has been research done. There have been research projects done on them to show their effectiveness, their efficacy, or their dangers, or this, that, the other. Well, as that information is out there and the AIs are starting to conglomerate human information, those AIs are going to present that information to these people who are working for the pharmaceuticals, and then they're going to see, and they, I believe, have seen, hey, there's a lot of efficacy here, there's a lot of use here, and that maybe this ought to be something that we should be looking at. So as they've brought that in and said, well, let's look at these GLP-1s, and then we see what that is, how that has changed our society with GLP-1s. And there's GLP-1 is a glucose-like peptide. It looks like a glucose, but it doesn't have any of the side effects of a glucose. There's no calories to it, but it tells cells that they're satiated. And so a GLP-1 is about communication from the gut, the liver, the pancreas, and the microbiome to the brain. And then we have GIP, which is it's a gastrointestinal peptide, which is about gut to gut, or gut microbiome with the gut information. So more and more information is coming out on these, and I do think the AI is having a much more powerful effect on bringing that out. And so now the pharmaceuticals are horning in on the peptide research. So, for example, one of the things on everyone's minds these days are some of these disease processes like Alzheimer's, dementia, et cetera, like brain processing and brain health has become a huge area of concern and interest. We're all interested in living longer, but also if we're going to live longer, we want to have a healthy, useful brain to enjoy
Quality
those years.
too, not just
not just
quantity of years, but also quality of life.
Quality and also being a contributing member to society, making our own lives more worthwhile. And so in that pursuit, we've gone off in different directions, and we've even talked about those in some of the other podcasts about the gut, the parabiosis, where they connected an old mouse and a young mouse, and trying to figure out, like, why did the old mouse's brain regenerate? Why did its organs regenerate? How do we find what it is that caused that? And so, of course, in that arena, it was kind of a dead end so far as pharmaceutical companies were concerned. So they've continued on saying, hey, that's nice, but there has to be something else that we can sell, something we can give people that will really have a strong effect. And hopefully, even once symptoms have shown up. So some of the other interventions where if you do it before the symptoms have shown up, then you have a much greater chance of keeping the symptoms from coming on. But what about when somebody has that problem? So one of the new things that has come out is the use of arginine and arginine-rich peptides. Okay, so arginine is it's a conditionally essential amino acid, which means that endogenous production or production by the body, it may be insufficient for our needs during periods of stress, injury, inflammation, or illness. So arginine.
So when we're having trouble, our body just can't produce enough of it.
Right. We need
to help
more
us
of
out.
that. Exactly.
Okay.
So there's some really interesting things that arginine does for us. So arginine just being an amino acid, and amino acids are the building blocks of peptides or proteins. So a peptide is a short chain fatty acid or a short chain protein, if you will. Protein is usually at least over 100 amino acids, and most peptides are 100 or less. Of course, there's some gray area into there. Is what are you going to call? We have some very large complexes that are peptides. They're partial proteins. Let's call it that. And that kind of leaves it open for where exactly that is. So one of the things that we found, and let me share this with you. There was a research article that basically, let's see if I can. Okay. I'm going to show you that. Okay, so here it is. Arginine and arginine-rich peptides as modulators of protein aggregation and cytotoxicity associated with Alzheimer's disease. So just a quick recap here. A substantial body of evidence indicates cationic arginine-rich peptides. Cationic just meaning it has a charge. Arginine-rich peptides. So a peptide, lots of amino acids. Arginine is one of the amino acids. As long as there's a lot of arginines in that peptide, it may have our effective therapeutic compounds for a range of neurodegenerative pathologies with beneficial effects, including the reduction of excitotoxic cell death and mitochondrial dysfunction. Now, mitochondria are the powerhouses or the power producing portions of our cells. So your neuron can be alive and not damaged, but if it doesn't have energy to run, it can't can't perform its function.
It's not going to be able to function
right,
properly.
exactly.
Sure.
So you can see how it's like, so excitotoxic, let's say that your neuron is working, and it wears out, like oxidative stress or excitation stress. It's used too many times and it wears the cell out and the cell dies. So those are the two things we're kind of talking about. So again, cationic arginine-rich peptides or CARPs therefore represent an emergent class of promising neurotherapeutics with multimodal mechanisms of action. So let's talk about that. So arginine.
So multimodal means that it works through different avenues,
different methods, right? There's multiple
different methods,
directions it comes from.
Mm-hmm.
Arginine itself is known as a chaotrope, able to prevent misfolding and aggregation of proteins. So let's a little background here. So amino acids, let's say you have a messenger RNA comes from the cell nucleus and says, I want you to build this protein. And that messenger RNA is then connected to a ribosome. And the ribosome says, perfect, I will just go through and click. I will read through this messenger RNA and I will follow the instructions for the peptide that it says to build. And as those amino acids are being added on the other side of the ribosome, it'll add, guanine, cytosine, et cetera. As those are being added on, you get a longer and longer string of amino acids. And as we said, that a peptide is anywhere from 2 to 50 or maybe a few more amino acids. And the reason for that is as this string of amino acids gets longer, all of a sudden there starts being these little folds in the structure. And initially we get primary folding and even secondary folding. And those, are fairly meaningless in and of themselves. But by the time the chain of amino acids starts to get longer, it starts to have what we call tertiary folding. And by the time we have that tertiary folding, all of a sudden this complex structure, that structure has a function. It causes an effect or can interact with other things that will cause an effect. For example, the cell wall or the cell nucleus or other proteins in the cell wall that will then cause another reaction somewhere else. So
is that just by the nature of its shape, it can have that effect on its surroundings, not necessarily what it contains, but just how it's formed. Okay.
That is how it has its effect, right? Is its shape. It is the three-dimensional shape. And there are some other aspects of the folding. And then what will happen is, let's say this protein's floating along through the body and it comes in contact with another protein. It causes that protein to change its shape, and it itself may change its shape. May or may not, depending on, and that's back to that multimodal function. These arginine-rich peptides, depending on their folding, they will have actions on various proteins throughout the body. Okay, so the arginine itself is known as chaotrope, able to prevent misfolding. So one of the things that will happen is as this amino acid chain is folding, there may be a misfold. There may be some other chemicals in the area or super free radical that causes a misfolding because an electron is added or another chemical structure is added. But interestingly enough, arginine may help any misfolding. So if you come out with the improper folding of a protein, it doesn't have its function. And especially as we age, that's more likely to happen. And if that misfolding happens in a neuron, the neuron doesn't function to its greatest potential. And so it may be kind of turned off or turned down and not able to function as high. Or so, for example, there's a thing called biofilms. Okay. There we go. There's my biofilm. So this red dotted line in this, can you see my cursor move, Brent?
I can see it. Yes, it's going.
Oh, okay.
Yeah.
Perfect. So a biofilm is oftentimes a collection of bacteria that with proteoglycans, et cetera. So basically mucus and bacteria. So bacteria make biofilms, but also the human body makes biofilms, and that's not really talked about as much. It's harder to find in the literature, but even the human body protects itself, and that's part of our microbiome. Like say the microbiome in the GI tract, or the microbiome in the genital tract, or in your mouth. These biofilms actually help protect us from organisms coming in. So our body produces mucus, and then that mucus interacts with these microorganisms that live there and live with the mucus, and they create a biofilm film that protects surfaces in our body from dust, pollen, other organisms coming in, chemicals that might come in and cause a problem. And they coat our lungs, coat our genitourinary tract, coat our GI tract. And when they're working properly, that they protect us. However, sometimes what happens is the microbiome, the organisms that are there get out of the right ratio, and they produce a biofilm that is not advantageous to us. It either gets too thick or it has the wrong organisms in it. And then that biofilm stops our bodily processes from happening appropriately. One of the classics is if you have an ulcer in your stomach, you might get a biofilm produced by Helicobacter pylori that then coats over that ulcer and won't let it heal. And so then you have a chronic ulcer. And so then we will give people antibiotics to say, hey, we got to stop that biofilm. So you can see that, okay, so now let's talk about another instance, since we're talking about Alzheimer's here. There is some consideration that we see amyloid beta and amyloid tau. Those are the misfolded proteins and the problematic structures that are in the brain that we've associated with Alzheimer's. And they are a biofilm of a sort. And some groups think that is in response to herpes simplex 1, the cold sore, or herpes simplex 2, the genital cold sore. And those can be up. And so some of these researchers believe that those organisms, that virus, makes its way eventually up into your brain area and causes this reaction of the misfolding proteins. And thus you get this biofilm.
This biofilm. Is that referred to in common literature as plaque? Is that the
plaque is a biofilm? Exactly. Okay. So in the mouth.
Right. I mean, like in the brain, because I've heard, in Alzheimer's discussions about a plaque that's in your brain that kind of prevents your neurons from connecting to each other or something like that. Is that
exactly? Well, okay. It will stop the connections and the growth of the neurons, so they won't make new, like for example, setting down new memories. And neurons interact with neurons in a new way, like for example, short-term memory, right? So how many people with Alzheimer's may have long-term memory, but they may not be able to have what was I doing 5 minutes ago, or what was it? Where am I going? Where am I driving to? So the short-term memory and the short-term function may be off, and that may be because of this biofilm. In fact, we've kind of associated Alzheimer's with amyloid beta, amyloid tau, and this biofilm. And
that's what I've heard in the past.
Yeah, that's fairly common knowledge. The herpes simplex virus, HSV1 and HSV2, being implicators of that. Like I said, some researchers have come to that conclusion, not all. But even getting the influenza, is another way that might occur, or other infective organisms, if you will, might cause that biofilm. But we get that biofilm, and now all of a sudden, for example, the other thing that makes our neurons work so well is that we put a myelin sheath around the neurons, so that makes them faster. So it's a little fat sheath. So what if we get a demyelination? And one of the ways you might get a demyelination is that when your body is trying to, replace myelin that has worn off or worn down or broken down, so you've got an axon, and then you have these little sheaths that go around it. And so what happens is, let's say that you have a long axon, and everywhere you have a sheath, the information will come, and then it jumps, and then it can go along, and then it will jump wherever those sheaths are. Well, let's say that your sheath is really large, so that the information on the neuron can come to here, and then it jumps all the way to here and fires. You make your neuron much faster. So you think about how some people are quick on their feet, right? They can think very quickly. What if we slowed that process down so that it took seconds and seconds, and now the neurons are stodgy? We might consider that Alzheimer's or dementia or age-related dementia, for example.
Cognitive decline.
Yeah. Right. And so that would be a misfolding of proteins that also help put that myelin together or help that sheath function properly. So now back to arginine. Well, if arginine and arginine-rich peptides might be capable of doing something about that. And so that's what they investigated, and that's what we want to talk about. Okay, so these biofilms, they're something that naturally occurs. They can abnormally occur. Another reason they might occur is because you have what we call organs in your brain that actually need to know what's going on in the bloodstream. So they are actually exposed to your regular bloodstream. So those are places where chemicals or organisms might be able to come and cause an ill effect, if you will. And again, this abnormal protein folding or cause a biofilm so that the brain structure cannot tell what's going on in the body and can't tell the body and the endocrine system and the hormonal system to tell the body how to function. So you can see how this can all start to break down.
Okay, right, it's like a cascade kind of.
It's like a cascade, and it might start off small, but at some point somebody goes, gosh, Grandpa has Alzheimer's. Okay, so then back to these arginine and arginine-rich peptides. So, and we put an L in front of arginine when we say that we want it to go across the blood-brain barrier, right? So you'll hear of L-lysine or L-arginine. Well, lysine and arginine are just amino acids, but let's say that somebody is supplementing and they want it to have a function in their brain. Another example might be thyroid medication. But anyway, so L-arginine is a conditionally essential amino acid, meaning our endogenous production may be insufficient during periods of stress, injury, inflammation, or illness to meet our needs. It's a key building block for proteins and a precursor for several biologically critical molecules, most notably nitric oxide. And nitric oxide is an interesting gas that the human body actually makes. For example, one of the places we see it the most is in a capillary bed for a red blood cell. A red blood cell is a pretty large structure, but for it to be able to go through a capillary bed, the capillary is very tiny. Here comes this huge red blood cell and it can't get into the capillary bed. And so it's trying to get in there. If there is the production of a little bit of nitric oxide just in front of that, the capillary bed will open up and then close down behind the red blood cell. So the red blood cell can go through the capillary bed when normally a red blood cell is far too large to go through a capillary bed by the use of nitric oxide. Interestingly, our body produces that not only in the precapillary sphincters, but also in response to stretching. So if you when you stretch your muscles or you foam roll or you do some of these self-help treatments, you're actually trying to get your body to produce nitric oxide, which all of a sudden makes everything loosen up and open up and increases blood flow so that you heal, you feel better, and you improve your overall health. So you can see how this nitric oxide function can be a very important aspect of arginine and arginine-rich peptides. Okay, so we've got nitric oxide production. Arginine is converted into nitric oxide. A potent vasodilator is what we call that. Nitric oxide improves blood flow, regulates blood pressure, supports endothelial function. Endothelium is the lining of your arteries, lining of your capillaries, and enhances tissue oxygenation.
It does all kinds of great stuff.
Yeah, all sorts of great stuff. So, okay, now I'm going to pull this off. So, okay, let's go back to our... Okay, so we can see that it does all sorts of great stuff. included in that is not only the nitric oxide production, but the tissue repair. Okay. Tissue repair and healing supports cell division, collagen synthesis, and wound healing. Well, collagen is, a lot of people have collagen injected into their face, or they take collagen orally to help make themselves look more youthful, to maintain healthy skin, healthy connective tissue. So you can see that, again, arginine is a very important substance. Also, immune function. It's essential for T cell activation. T cells are our thymus cells, or our white blood cells that help fight infection. And it also assists in immune regulation, plays a role in the inflammatory balance. Can you turn inflammation on and then turn it off? Very important. It's the turning it off part that sometimes humans have a real hard time with. Hormone regulation stimulates release of growth hormone and insulin, kind of important, influencing metabolism and tissue regeneration. So again, how do you get somebody to be vibrant, to be young, to be healthy? If they don't have the proper amounts of arginine, especially when they've been stressed, for example, a stressor can be exercising at really high levels. Think of our Olympic athletes who then are all sick during the Olympics because they've been training so hard, and then when it's finally time for their event, they just, their immune system kind of falls apart, and their ability to regulate their immune system has kind of fallen apart.
Mm-hmm. Can I ask you a question real quick?
Sure.
So what are the precursors that the body uses to produce arginine, like, naturally?
Well, typically we ingest it, right? So
it's already found in the food that we, the food that we eat.
Exactly.
Yeah,
and that's what it's called, a conditional essential amino acid. So an essential amino acid is something that we need to consume in order to
survive, right?
To
Just
survive,
to live.
right? Yeah. Let's see, meat, poultry, and fish, dairy products, nuts, seed, soy, and whole grains are sources of arginine. Okay. So, those are all important, and you can see why actually the food food pyramid was recently kind of rejiggered by the Health and Human Services, really saying, look, more meat, more poultry, more fish, dairy products are important. And arginine would be one of the reasons that they would make that claim, saying, look, some of these essential amino acids are in this food. They're not in carbs and potato chips and some of these other things. Notice I didn't say anything about, there was no green vegetable, there was no carb, there was no pasta. Arginine's not in any of those things. So unless they happen to be made out of nuts, seeds, soy, or whole grains. Whole grains, I guess. Yeah, actually, I take that back. Whole grains. But the problem is, what is a whole grain? Well, as soon as you crush it, it's not a whole grain.
Right. The husk is damaged, right?
Yeah. It becomes damaged, and we start separating out the husk and making things. Well, if you can bake bread out of it, it's not a whole grain anymore, even though we say we have whole grain. Yeah, well, it started as whole grain, but that's one of the important distinctions. When you have food and then it has fiber, and it has soluble and insoluble fiber. Once you separate the soluble and insoluble fiber from each other, they no longer work as a unit. So, for example, when you eat a fruit, there's soluble and there's insoluble fiber. So let's say that the insoluble fiber is like a netting, and then the soluble fiber is like a gel that covers it. And then within that fruit is all this sugar, right? Fructose. Because the soluble and the insoluble and the soluble fiber coat your intestinal tract, that sugar cannot immediately absorb across the intestine wall. If it immediately absorbs across the intestine wall, you get really high spikes in your blood sugar. Causing diabetes, kind of a form of diabetes. You start to have glucose spikes. If that fiber is there, if those two forms of fiber are there, the substance moves past the initial portions of your small intestine, where now it's no longer able to cause a spike in blood sugar. Right. So now, even though you ingested all this fructose with your fruit, because it had the soluble and insoluble fiber, it won't cause blood sugar spikes, thus not inducing type 2 diabetes. That's, again, why the recommendations more recently about the food pyramid are whole fruits. And when we say whole grains, that's nice, but as soon as you crush it and mill it, it's no longer a whole grain. So to call something a whole grain when it's in a bread is misleading. You can't have whole grain in a bread so unless you're chewing on wheat, which none of us are going to do, it doesn't have the health benefits that we'd like to say Arginine is still in that, but it's not the same thing. So again, some of these terms we have to be more careful with, and it seems like we are getting better about going back and redefining these. I mean, this information was all changed in the 70s, and now we're kind of changing it back to more what was known, or what we said in the 70s with that. Anyway, moving on. So detoxification, participation in the urea cycle, assisting the kidneys and removing nitrogenous waste. So nitrogenous waste is one of the problems with eating a ton of protein is that it causes nitrogenous waste. There's lots of nitrogen in there, and that is processed by the kidneys. So if you do damage to the kidneys by putting all this nitrogenous waste through there, that's problematic because we don't want kidney disease. Well, guess what? Arginine helps break down, again, biofilms, and that maybe this nitrogen would help feed, right? Because obviously bacteria are going to be good at feeding off of nitrogen or nitrogenous-based ammonia. They're going to be good at feeding off that. So if that creates a biofilm, you'd want to be able to break that down. Again, arginine assists with that. Okay, so
Fascinating.
Yeah, so you can see that arginine is very important. And of course the supplementation. Now here's really where the rub comes in is like, look, as a osteopathic manual therapist, I'm working all the time to break down biofilms to make surfaces work again. Yeah, we don't always call it a biofilm, but it really is. And here I want to make sure that there it is. I'll share this with you in just a second.
No worries. I was just going to interject and say one of the things that Autumn likes to say, and she's quoted this as, I think it came from Bear, is that when she's working on muscles in particular, especially in the legs, she wants the muscles to be slicky, not sticky. Is that
exactly
one of those? Does that relate to breaking down biofilms as an osteopathic manual therapist?
Exactly, because mucus is a biofilm, right? And our body produces the substrate of that. However, our body has bacteria all throughout it. And so now sometimes, again, if those substances get in the wrong amount, for example, and what she's talking about there, slicky, not sticky, when you have proper autonomic tone, in other words, the parasympathetic and the sympathetic tone are even or appropriate, where they kind of vacillate back and forth, you have fluid that is viscous, but it's serous. So it's you get a serous fluid that still is kind of sticks together, but isn't too thick.
Mm-hmm.
Whereas if you have high sympathetic tone, you get too viscous and you get it sticky. So that's what she's saying, slicky, not sticky. Well, a sticky biofilm is partially produced because of high sympathetic tone, and then too serous, too watery, and that's caused by too high of parasympathetic tone. So both ends of that are bad, right?
Mm-hmm.
We really want this vacillation in the middle of on their toes, nimble and quick and responsive. So
are those related to biofilms at all? Did you say
those are biofilms? Right, exactly.
Those are
okay.
Those
biofilms.
fluids are biofilms. Okay.
I'm going to show you. I'm going to share this with you. Let me share this with you. Trying to maneuver this around here. Okay, here we go. Not that. This one here. There we go. So here are all the places that biofilms occur in the human body. Let's get that. Okay, there we go. So for example, in the middle ear, in the upper respiratory tract, the cardiovascular system, on your teeth, as you had mentioned, lung sputum, gastric mucosa. We were talking about the stomach ulcer, urogenital system, colonic mucosa, soft tissue wounds, in bones, and obviously on joints. Now, typically, what we're talking about when we're talking about those is that we say, oh, biofilm, and we mean the pejorative part where a bacteria is overgrown and we have an infection there. So you can get a bone infection, you can have a joint infection, you can have, especially once we've put a prosthesis in there or something like that, there's all sorts of things that will introduce something and it makes it much more likely that you'll get an infection there. However, you do have bacteria through your body. You do have viruses throughout your body. You have all sorts of organisms living in your body with you. So those biofilms are everywhere. And we use them to keep certain things out, but also allow other things to come in. So if they get too thick, as we said, as Autumn said, too sticky, then things can't get in. Now that's great if you have an infection, because now you stick all these bacteria that are overgrowing, you stick them together and say, you can't go anywhere. And then our immune system, our T cells, our white blood cells will come in and then destroy that area. And of course you'll cough it up or, blow it out, blow your nose or cough it up and get rid of it. And so that's what you're doing with a lot of that sputum. So, exactly with that biofilm. Well, what if that occurs in a joint? What do you do about that? So as an osteopathic manual therapist, I go through and I try to improve the motion of the joint, and making sure that, hey, can we break that down? What about the slicky part that's supposed to occur between muscles? What if that serous mucus layer, that biofilm, isn't there so that the muscles can slide on each other easily? Well, you get tendinitis, or if it wears long enough, tendinosis, or muscle strain, et cetera, et cetera. So heat builds up, oxygen. There's a loss of oxygen in the area. You don't have proper functioning of structures. They become very irritable, and so you might get muscle spasm or muscle problems, and leads it more likely that you would have a tear, et cetera, et cetera. So I personally, and a lot of practitioners, whether they know it or not, are working on biofilms. And so it's important to understand that you're working on biofilms. And you think about, let's talk about a time that a biofilm doesn't work for you. Let's say you have two pieces of glass and you put a drop of water between them, and the two pieces of glass get stuck together, and then you need these to come apart. Now this happens in other times where water adheres to structures together, and then you're like, I need those two to come apart, and they kind of pop apart, right? So we all have experienced this in some fashion, but we're, as a manual therapist, I'm dealing with that in the human body. So knowing this information about arginine and the brain, and like, for example, when you do craniosacral work, or when you work on a joint, or even when you're working on muscles or tendons or ligaments, anywhere that there's fluid that can either be too serous or too viscous, right? I'm trying to restore that biofilm to its normal function of being slicky, not sticky, but not watery, right? Not watery, not sticky. So that's what Autumn and our mentor Bear was referring to, is that, does that biofilm work appropriately? Is it an appropriate biofilm? The problem being is sometimes you get into conditions where, as a manual therapist, you have a really difficult time breaking that down. And for example, Alzheimer's, what do I do about Alzheimer's? Well, I might do cranial, I might do this, right? But you're going, that's going to be, not a lot, right?
Right. Because part of your job, it sounds like, with biofilm is to physically, break it up, I guess, through manipulation. Yeah. But also you have to get the body itself to regulate its production and its, I guess, I don't know, does the body have like its own removal mechanisms that maybe get overwhelmed and that causes a biofilm buildup?
Exactly. So the autonomic nervous system is overseeing, like, what are the chemicals and the peptides in an area? Like, what is the nutrition in an area? For example, the sympathetics tend to close down arteries, and the parasympathetics action, especially through nitric oxide, would be to vasodilate, or to produce more, to make a biofilm more permeable, and the sympathetics make it less permeable. Well, what if there's some nutrition, there's a biofilm, and you have nutrition on this side closer to the artery, but you need it in here in the capillary bed, and it can't get here because of this biofilm? Well, you're going to have aching. So for example, we might call that fibromyalgia. We might call that tendinitis. We might call that whatever problem there might be. This barrier is now keeping us from getting nutrients that are being driven along by the sympathetics and the parasympathetics. You just can't get it to where you need it to go. So one of the things that the body does is says, sometimes I need to break down a biofilm. So it takes the arginine that it has available to it through nutrition and says, hey, I've got some compounds that are arginine-rich peptides, and I will use those to help connect that. But as we've seen, like with aging and with exposure to chemicals, or just our lives, like what we do, say somebody's working all the time, and they never give themselves a break, and so they might develop Alzheimer's, or they might have brain fog, or et cetera, or we take chemo, right? We will create these biofilms, or we have too much stress, and so everything's become sticky and not slicky, and so we really lose our ability to control the environment, and by controlling that environment, we induce health in the cells. And so it's very important for us to have not only proper arginine supplementation, but you still have to change the autonomic control, right, to be able to get the arginine there. And that's, I guess, where I feel like I come in and saying, hey, look, not only is this important, but you also have to help the body get there, because you can have all the arginine you want in your stomach, but if you can't absorb it, process it through the liver, get it over to where it needs to be, and get it to where the biofilm is, and then mechanically, biochemically, bioelectrically, right? That's what OsteoSignal really means, is what are the signals of health of the human body? Biomechanical function, biochemical function, and bioelectrical function. And arginine is one of those things that will help break down biofilms that have become too sticky, and help break that down so that we can, again, get back to that. I help that through inducing changing blood flow, changing how nutrition is able to move through the system, getting rid of, helping people, helping people's body move lymphedema or lymph along. Right, if the lymph is in the area and it's gotten thickened all up, it becomes a really thick biofilm. In fact, it becomes just a biogel at that point, and now there's no breaking through it. So my job is to help clear the way and make the processing, what are the structures in the human body that are designed to function with this? My job is to make that possible. But sometimes breaking down this biofilm can be very, very difficult. And what my efforts might be getting it at the edges, but the arginine-rich peptides will really put big holes in it and allow it to really change how it's functioning quickly. And that's the whole idea. And again, that's a natural process already occurring in the body. Some people need more, particularly people with Alzheimer's or with other biofilm problems. So that's the other part I guess I'm suggesting here, is that we need to not only know what biofilm problems are, but be able to recognize them and then understand, hey, maybe this person has a higher need for arginine than another person, and suggest that maybe you should look at arginine supplementation, or go talk to your naturopath, go talk to your doctor, go speak to somebody. But here's what the research says. It says arginine and arginine-rich peptides are very helpful for this. And I can see that you have a biofilm, or a biogel at this point. A gel is just a really thick film that we're having a real difficult time breaking down. And again, even in medical implantation is one of the big areas that you get infection in biofilms. So for example, I have a patient's type 1 diabetic, and she's had so many insulin sites, pump sites. So she has an insulin pump, and she has to put a little catheter inside of her skin, and she gets a biofilm on that within 3 days. And so it stops the function of the like, for example, she says, oh, my insulin's bad. It's like, well, it's that really the insulin can't get in because the biofilm is just blocking the insulin flow in the area. Right. An everyday occurrence for this person. And so she has to move her pump site around. And of course, at some point you get older, she's 62 now. So she's running out of sites. You don't want to walk around with an insulin site on your forehead. So she's running out of sites to put this. So breaking down these biofilms and doing something about that, but also understanding, like, hey, one of the things that can really help us with this, I can do that biomechanically, biochemically, bioelectrically, but I can also have you take arginine or arginine-rich peptides that will really help us break those down and improve the function of your medical device, improve the function of your prosthetic knee, or your arthroplastic knee, or just improve the function of the joints that you already have, or the tissues that you already have where you might have a problem. People with chronic cough or chronic lung problems, heart problems, cardiovascular problems. We're saying this biofilm thing, and then we start saying, well, like plaques. There's some level of, arterial sclerosis or plaques in the arteries. Aren't we speaking about a similar process? Yes, we are. So there's not been research done on that particularly, but again, that's why we're having this conversation, to talk about what are the things that we want to think about, like, hey, what might this help with, and why should people make sure that they're getting proper arginine supplementation? And again, it might just be in the food they're taking, but it can also be something very effective to add to their diet.
For sure. I just wanted to ask a quick question about biofilm breakdown. Is autophagy part of that process, or is that a different mechanism for getting rid of other waste?
Well, so we're talking about cells, and autophagy is really a breakdown of we're trying to cause auto cell lysis of cells that are maybe older, so that younger cells will grow in their place. And so we're trying to turn those over. But that autophagy, when you go to do that, will lend itself to the biofilm, because now you have broken down
because the cells break down and you have more cell waste. So
and
it's
you
a
have
different
to move that out to the lymphatic channels. And if they get backed up, again, you get a thickened biofilm and a biogel at some point, and now the area, and you're going, gosh, I'm really trying for this autophagy, and I'm not getting the results I expected. It may
So it's
be
a
that you need,
different, it's a different process in the body. It sounds like breaking down biofilm and like getting rid of old cells are two different kind of
well,
things.
they are and they
Like,
aren't, because the biofilms are such a part of how we process everything. How does our lymphatics
mhm.
work? How do our arteries and our veins work? How do our joints work? So this is on many different levels. And again, the lymphatic flow, you can get lymph stasis from that. Or, so for example, we're trying to get autophagy because you're saying, look, something's not going right. That cell probably should have died already, or I should have new, younger cells in place. But for whatever reason, that process isn't happening. So I'm going to do this other thing that causes autophagy, whether that's, uh, you know, not eating for a long time, or working out a certain way, or working out in a really hot environment, whatever you're trying to do to get cell turnover. The problem is that the cell turnover itself is also dependent upon, do those cells, are they able to go through with that process of autophagy? Because if they're not getting the right signals, if they're not getting bathed in cerebrospinal fluid appropriately, if they're not getting signaling from cerebrospinal fluid appropriately, if they're not getting hormone signaling, right, because of this biofilm, then they're not going to get those signals to go through the process of autophagy. So they're just going to sit there, not particularly functioning correctly, but not dying. They need that clear signal of, oh, it's my time to die. I'll die. And then that refuse has to be broken down, recycled, and taken away. So it really does, the process of autophagy is creating a biofilm.
That's fascinating. That's really cool. And then, so how does the body process biofilm that's been broken down? Does it go, I think, did you mention it goes to the kidneys? Is that,
Well,
am I understanding that correctly? Or how does it get removed
yeah.
from the body?
So your any broken down substances like that move through your lymphatic system, and your lymphatic system dumps them back into your, well, actually, yeah, 3/4 of your body dumps into your left thoracic duct and back into your heart, actually. So your lymphatic system collects and then comes up and dumps itself back into your bloodstream. And then in the bloodstream, the different parts of the biofilm are going to be, of course, still being broken down. They're going to be, they're going to be attacked by, or processed by white blood cells, macrophages. Your kidneys are going to have some portion of that, particularly nitrogenous waste. Nitrogenous waste, anything with nitrogen in it, protein. So autophagy, a lot of times that is also part of an exercise program. And so you're going to break down muscle. Well, when you break down that muscle, and those amino acids that make up that protein have to be processed. That processing, now your system is going to is going to kind of recycle some of that. But at some point, your system says, whoa, too much stuff, let's just dump it. So your kidneys are concerned mostly with getting rid of that.
Okay, awesome.
So what they need to do is they need to take the nitrogen and attach it so that it can go through without destroying the place.
Mhm.
So, yeah. So
Okay,
protein
cool.
metabolism is about the kidneys. Now, there's other parts of the biofilm that are not protein. They might be carbohydrate or fats, cholesterols. Those might be processed more by the colon.
Gotcha. Okay.
Yeah.
Awesome.
So then that's
Thanks.
how the whole system is supposed to keep itself in balance and keep moving, regenerate, renew, rejuvenate. And, but you can see how biofilms are supposed to assist that. But especially as we age and we come in contact with things, or we do the same processes over and over, the same, postural adaptation, that we might cause a biofilm that becomes a biogel, and things don't work quite like they're supposed to. Okay. So that hopefully is helpful. So, and one of the things we want to do is these arginine-rich peptides, again, so they have a cell penetrating ability, right? So they can go into the cell and cause your DNA, like if there's a signal your DNA and the cell nucleus needs to get, or the cell itself, like for example, we were talking about autophagy, arginine-rich peptides will get that signal into the cell. They'll get it into the nucleus, so that the DNA, how we open up our DNA and cause proteins to be made or other structures to be made, we can improve that. Also, membrane, as we said, membrane modulation, induce membrane rearrangement, fusion, or uptake via macropinocytosis. So that just means that arginine-rich peptides can be a large molecule, and so what will happen is a cell will make a little vacuole, and then it will like break down some of it and suck little bits into the cell and make use of it. So there's all sorts of ways that this is that it helps the human body also intracellular delivery, for example, medications or drugs, or anything that medically we might want to get it into the cell. One thing that this is going to be helpful for is cancer cells. Cancer cells are also going to use the same process because they're somewhat normal cells with some slight problems with them. Well, let's say we wanted chemo to get into that cancer cell. If you don't have enough arginine, that chemo is not going to get into that cell. So you can see the wide range of uses of arginine, but also the importance of understanding a biofilm, because what happens with cancers is that first off, your body builds a biofilm around it, and then the cancer itself has its own biofilm, right? Because it doesn't want things from outside. So we may not have as effective of chemo. The chemo may not be as effective because tumors particularly are always trying to get their own blood flow. They're trying to get blood flow because they have poor blood flow. So we're, giving a person chemo. There's these two biofilms, one your body's, one the cancer's, that are blocking the chemo from getting in there. But if you're taking arginine or arginine-rich peptides, that will put holes in there and then help deliver this right into the cancer cell. So again, further implications for why this is so necessary. But also,
yeah,
go ahead.
Sorry, I was just going to kind of circle back to Alzheimer's.
Yes.
So that arginine, I think maybe we need to talk a little bit more about what it does with the biofilm in the, in the brain.
In the brain, yeah. So
could you elaborate on that? How does that keep the brain healthy and let people, keep their cognitive function longer?
Yeah. So let's go back to those into the cell. So let's say that this arginine helps the cell have proper signaling. So the biofilm can change the chemistry in the area. It'll change the pH. So the arginine will change the pH. And when you change pH and chemical processes, you actually change which direction the chemical process goes. If you change the concentration gradients, like how much of a certain chemical is in an area, if there's too much and it can't get out, then that will stop chemical processes. So the way that arginine is going to help Alzheimer's, it is by breaking down that biofilm, that amyloid beta, that amyloid tau that we're seeing in the brain. It will break that down and allow the cells to have the proper chemistry. And by having proper chemistry, for example, changes in pH. If an area is contained in a biofilm, it may not be able to change its pH. It may not be able to move chemicals in and out. So there's also concentration gradients. So there's pH and there's concentration gradients. And those things both affect chemical processes, growth processes. So now you have these brain cells that can't go through their normal chemical processing, or they're bathed in chemicals they shouldn't be bathed in, so that it's going to cause them to misfold their proteins when they're making proteins inside, or the support cells with them, the astrocytes, the Schwann cells, etc., the dendritic cells that really help neurons do their job. They may have this problem as well. And so the chemistry of the area will be off. And the biofilm maintains that. An improper biofilm maintains that. So this is where the arginine coming in and putting holes in that and keeping the biofilm slicky, not sticky, not too serous, not too thick, not too viscous, but just right to allow certain chemicals in and out, proteins in and out, hormones in and out. Then those cells will be able to make proteins, make energy. Their mitochondria will be able to function appropriately. And so, for example, waste products, the extracellular space around a neuron has to be cleared of all sorts of waste products. And again, some of these chemicals, they build up too high and that has to be moved out. But if you have a biofilm there, that can't happen. So that's where arginine comes in, and it comes in and breaks down those biofilms, keeps them from being too stable and being more responsive to the area chemically.
And it lets those bioelectrical signals flow without too much impedance. Yeah.
Right. And so then that way the neuron is able to communicate appropriately. It gets the proper chemistry so it can make proteins, keep itself healthy. The support cells can keep it healthy. But also then the signaling for the arteries and the arterial support. Of course, there's the blood-brain barrier there, again, the support cells. But that whole area, you will have appropriate signaling all the way through, because the blood-brain barrier, in a way, is a biofilm. you have these cells that are extra wrapping the arteries and keeping the chemical stuff in the artery from affecting the brain cells too much. But if that biofilm gets too thick, then you can't get enough nutrients in, or enough oxygen in, or enough substrates in. And then again, the cell can't do its job. The support cells can't do their job. They can't keep the neuron healthy. And so you get a neuron that doesn't fire very often, doesn't fire when it should, is hyperreactive, hyporeactive, or misfolds its proteins and becomes kind of broken down, if you will. It's more of a jalopy as opposed to a sports car.
Sure. All right.
Okay, so that's
all
really the benefit of
fascinating.
that. Yeah, and that's what we're trying to do with osteopathic manual therapy. We do craniosacral. We're trying to get the tissues of the skull and the support structures to function properly. But if there's a biofilm that we just can't break down, right, because there's only so much you can do to the skull, then this would be very useful.
All right. I don't know if you have time to maybe describe a technique or two. We can always edit this out, but it would be,
sure.
And it might give listeners, kind of an idea of exactly how you would go about, promoting the healthy use of arginine in the body, or promote healthy signaling to use arginine in the body. Can you maybe describe like a technique that helps support that?
I mean, like a manual technique that we do.
If there's not time, that's fine. But I think it might just to help people listening, visualize, what would that look like? How would you actually accomplish that in the skull or in the, you said craniosacral, so there's a link between the cranium and the sacrum, right? Is that? Yeah.
So, and the link is this: that you have this bag around your central nervous system. It encompasses your brain all the way down your spinal cord and all the way to your tailbone, in fact. And so the movement of the sacrum and the movement of the cranium, even though it's more just flexing than true movement, causes a fluxing of fluids in there. And one of those fluids is cerebrospinal fluid. So the production of the cerebrospinal fluid occurs in the choroid plexuses, but that fluid needs to be moved through. There has to be a current of this fluid through the brain, the spinal cord, all the way down to the coccyx. And then that cerebrospinal fluid is supposed to weep out of the skull through the lymphatics and drain in and signal all the cells of the body. And 2/3 of that happens in the cranium and 1/3 in the sacrum, right? So a very important process, not well known or well understood. But so that what we call the dural membrane, or that dura, that sac, it also makes divisions in the brain itself. So it actually divides the lobes of the brain in some different areas. And craniosacral is kind of based on making not only the bones move, because they are adhered to the dural sac, but also there's a double thickening called the falx cerebri and the tentorium cerebelli, okay, that are attached to these bones specifically. And it's what we call a reciprocal tension membrane. In other words, that as the skull goes through its natural deformation, a rhythmic deformation, to help pump these fluids, the cerebrospinal fluid, this double sac, this dural membrane, is what is actually acting as a bellows. Well, there can be some real problems with that. The bellows might be non-functioning or not functioning optimally because of, say, a birth injury, that as the baby came through the birth canal, the brain case might have gotten squished off to the side. And then, babies are supposed to cry and this, that, the other, and their heads are supposed to pop back out. And people sometimes say, oh, look, my baby's head looks funny. We're going to use this helmet to help push the bones back into place. Unfortunately, the helmets, they make the shape look right, but they don't restore the function of the dural membrane. So the important part here is the methods that I use are to go after that dural membrane and say, can that dural membrane go through its mild flexing? And so on the sides of the brain, it's doing this, right? And then at the front to back are doing this. So one of the problems we see with babies is they get stuck. They get what's called a scallop head, right? Their head gets stuck because those sutures close prematurely and their head does not move forward and backward anymore, and they get a scallop head. You've also, conversely, if it gets stuck on the sides, you've seen that baby whose head looks like a bowling ball and it's really flat front to back, but very wide on the sides. So those are two problems that'll occur with the skull. Now, those are really bad problems, but then there are lesser degrees of that all the way to pretty normal. And most of us have pretty normal. We don't have a huge problem, but you can maximize the function of that movement of that cerebral spinal fluid across the central nervous system by making these dural membranes, allowing these dural membranes to flex appropriately.
To flex, yeah. Is the dura, does it meet the definition of a biofilm?
It's a hard film.
Okay.
That is, that is, for example, if someone cracks their skull and then like a clear fluid leaks out, like some people hit their head and then blood and a little clear fluid will come out, we will know that they've cracked their skull or fractured their skull, and people can die from that.
Yeah.
It is a very rigid, so the outer part is not a biofilm. However, on the inside, and particularly around the arteries, and here's the fun part, this sac does double over, right? It'll double over, and in the doubling, that's where all the veins are. So it is a biofilm. It acts as a biofilm inside the skull in certain parts of it. And that's the important part to understand.
All right.
Yeah.
I was just trying to tie all those things together.
Yes, exactly.
Yeah.
So, and then that movement of that cerebral spinal fluid. So the inner layer, so there's the dura, the arachnoid, and the pia mater. So the pia mater is adhered to the brain, so it's definitely a biofilm. And then the arachnoid space, which is where we get a subdural hematoma, right? We talk about the dural membrane. Below the dura in the arachnoid space can be a biofilm as well. And so the inside of that dura is a biofilm. So we have lots of layers of areas where we would have a biofilm to protect the brain from infection, particularly. And so, my job as a craniosacral, when I do craniosacral, is to maximize the function of that so that the biofilm functions as best as I can make it. But again, back to the arginine-rich peptides, if you're having a particular problem with that, an advanced problem with that, just doing cranial is not necessarily going to reverse that process because it's such an advanced problem. And this is where having a supplementation or something like that. And again, like I said, the pharmaceuticals will be coming out with something soon based on this research.
Gotcha.
You can be
And
guaranteed.
it's always a good idea. Yeah. It's always a good idea to, if you're going to supplement with anything, do your homework, talk to a nutritionist or doctor.
Yeah, exactly.
Right.
Naturopath, somebody who's qualified to make those, those determinations. Because there's certainly, there can be some people who have to watch their arginine, some MTHFR people, some people with other, things. So again, you medically, you always have to go. And like I said, arginine at this point is considered a nutraceutical. But at some point, the pharmaceuticals will have a pharmaceutical version, just like a red yeast rice is the nutraceutical version of a statin. And again, nutraceuticals, like you said, a nutritionist, definitely do your own reading, talk to your pharmacist. There's all sorts of places where you can get good information on, hey, is this something that would be appropriate for me? Is this something that would be harmful to me? Especially if you're taking other medications. Your pharmacist is almost always the best at knowing what are the drug interactions. So that's, those are always people you should talk to saying, hey, I'm taking this and this and this. And then you don't know it, but I'm taking this nutraceutical, an arginine-rich peptide. So I want to make sure that doesn't interfere with anything. So yeah, very good advice to make sure people get good information and make sure it's appropriate for them. But it can be very useful. And particularly just because you're using, and I guess going back, if you have inflammation and you have other factors that would not allow the arginine to get where it needs to go, you also need that osteopathic manual therapy to help make the system function to maximize the value of the nutrition, of the nutraceutical in this. And in fact, there's, for example, one thing is I have people who will be on blood pressure medication. And of course, they're on this much blood pressure medication. And I'll start working with them. And then pretty soon they're getting dizzy all the time. And I have to tell them, hey, you need to go back to your cardiologist, tell them that we've made some changes and that now that level of medication is making you dizzy all the time. And then the doctor will say, well, let's rejigger this, and they'll do this, or they might change the medication or whatever. But the doctor will determine, let's go at a lower dose. And then that ends up being just as effective because now we've made their body work better. So now they won't have as high blood pressure or other problems. So yes, you can see how this interacts, how the manual therapy interacts with supplementation, pharmaceutical, nutraceutical. But again, proper information.
For sure. This was a fascinating discussion. I didn't even know that biofilms existed. I mean, well, you see them, in your teeth or
Yeah,
whatever,
yeah, yeah.
mucus, whatever, but just how the role that they play with all the different systems in your body, like that graphic you showed of where biofilms occur in different parts of the body,
Right, exactly.
right?
Exactly.
And the problems with, if they get too thick, that can be really problematic. Is there a problem if they, I mean, yeah, and I guess if they're not present, then you also can also have certain problems, right? If you don't have
Exactly.
biofilms,
Oh, no,
where they're
exactly.
supposed
Then
to
you
be.
would get infections. Oh, this is the wrong one. This is the one. I was going to show that one more time, but apparently not.
Okay, yeah. So that article is from some place in Western Australia, it looks like, from what I could tell. And
Yes.
so, yeah,
Let's see where these researchers are from.
we'll put maybe we could put a link in the description
Yes, I'll send that
so
to
that
you.
people can also find that out. Would you be?
If I have the right one or not.
Yeah.
Okay, so they were from Molecular Sciences, Faculty of Science, University of Western Australia. Yeah, all Western Australia.
Yeah. Okay. Yeah, it's such a cool thing
Yes.
to kind of dig deep into and just see how these peptides, arginine-rich peptides, can help make sure that your biofilms, especially in your brain, right,
Exactly.
are at the adequate levels to make sure that you're, maybe I was going to ask you, what did they discover with the effects on Alzheimer's? Did it show a
Yeah,
certain
let's
result?
read
Did
that.
you have time to talk about that?
Yeah, let's pull that up. That's the one. Yes. Oh, nope, that's not it.
Because we talked about the theory behind it and what it's supposed to do, but I wonder if
Yeah,
this
let's see what
paper
they.
mentioned, their findings. How did it affect people suffering from Alzheimer's or dementia?
Okay, I seem to have lost it. Hold on.
Oh, no worries. So that was their thesis, right? That these carps.
Yes. Okay, there we go. It's on here. It's this one. It's this one. Okay, yes, these carps would help with that. Okay, so let's go down to their discussion all the way at the bottom here. Okay, so their conclusion, arginine has several distinctive properties, largely owing to the unique structure and chemistry of the guanidinium group. While monomeric arginine has long been regarded as a potent inhibitor of protein aggregation, polypeptides enriched for arginine residues also display interesting effects on the formation and cytotoxicity of protein aggregates. So when proteins can glom up in a ball, they can have negative effects in an area, partially because they have electrical charge, partially because of their chemical effects. And we talked about pH. So we can have interesting effects on the formation and cytotoxicity of protein aggregates. Arginine residues have substantial bioactive properties in the context of modulating protein aggregation and should therefore be given particular consideration in the rational design of amyloid targeting therapeutic peptides. And again, because amyloid beta and tau are proteins that aggregate in an area, and so their thought process was that it would. Okay, so that so it would have an effect on this. So interestingly, carps have also demonstrated significant neuroprotective effects in models of stroke and traumatic brain injury, particularly by targeting excitotoxicity, which is also a pathological feature of Alzheimer's disease. Okay, the complex and multifactorial nature of Alzheimer's pathogenesis particularly warrants therapeutic candidates with multiple mechanisms of action. Okay.
So it seems like it's saying that Alzheimer's shares some pathological features with other, brain ailments
Injuries, like, for example, stroke.
that these carps have been able to help ameliorate. And so it's interesting to look at. So it sounds like they're saying that this would help address one aspect of Alzheimer's, and that there are maybe some other aspects that other treatment methods would help. Am I getting that
Exactly.
right? Okay.
Exactly. So if we go after the amyloid beta part of it, the protein aggregation, and a protein aggregation, for example, in a stroke, would be that that proteins are going to aggregate. For example, like blood has leaked into an area in the brain, and those are a bunch of proteins, and the protein draws in a lot of fluid. That's one of the big problems with protein aggregation. So that biofilm also pulls in a ton of fluid. Well, in the brain, a lot of fluid is going to put a lot of pressure on your brain, and that pressure on the brain is obviously going to suppress its function and kill cells. So by breaking down that biofilm and undermining protein aggregation, now all of a sudden the fluid moves out and you don't have the pressure on the brain. And the same occurs in a stroke. Similar thing with the amyloid beta and the amyloid tau. Traumatic brain injury was the other example that they used. So you can see that, yes, that this is one method. And, in medical stuff, we try not to step on everyone's toes. They're not trying to say, hey, this is going to replace everything. What they're trying to say is, hey, let's look further into this. Let's start doing clinical trials. We've seen this have this effect, but we would like to do full-on clinical trials with that.
Sure. All right. Awesome. That's great.
Yeah. And like I said, the other aspects of people's lives and their function that this is going to affect. There we go. We're back.
All right.
Okay. Yeah, that sounds like we got all the way there. I did just want to say that arginine is clinically relevant amino acid supporting circulation, immune function, and tissue repair. Arginine-rich peptides represent a frontier in molecular medicine, offering insights into how cellular communication and repair can be influenced beyond mechanical intervention. Okay, and which, while not directly applied in osteopathic manual therapy, peptide biology strengthens the scientific foundation underlying osteopathic principles. Is again about the biofilms and understanding how we're working with the body and how it functions and where it falls out of proper function either direction, whether too sticky, too slicky. So it is really useful to use this to look into how do we make bodies work better.
Awesome. And definitely osteopathic manual therapy plays a role in that, as you
Yes.
detailed. So yeah,
Yeah,
I
our
really
job
appreciate that.
to improve the biomechanical, the biochemical, and the bioelectrical functioning and signaling of the body.
All right. Thanks, Neal. Really appreciate it. And thanks to all you joining us for OsteoSignal with Neal O'Neill. And we'll see you next time.
See you next time.
Thanks for joining us on another episode of OsteoSignal with Neal D. O'Neill. For more information, please visit pursuitpt.com or pursuitperformancetraining.com. Our music was written and performed by Sky Toes. Copyright 2026 by Neal D. O'Neill.